to BioLibrary, and today we'll start with chaperones. I've already told you enough about this concept. I think if you haven't watched the previous videos of mine, please go and watch. Definitely you're gonna get some perspective on chaperones and as to why are there and what is their function in practical means. Okay, so let's start with this video. This video contains notes from Leninger as well as my notes, so it will help you to get through the questions of chaperones for any exam, like be it your CSIR, be it GATE, be it your BARC exam. In fact, GATE doesn't ask this level of questions, but then you can attempt the questions in DBT, all one day exams of your different institutes for PhD. Okay, so let's start with the video. So let's start, everybody. And first of all, this is let's define chaperone. Chaperones, why are they there in the first place? Because there are proteins which are partly folded or maybe in infolded form, or sorry, maybe in unfolded form, or they are improperly folded. So to handle them, there are a different class of proteins present in your cell called as chaperones. As the first line suggests, that folding for many proteins requires chaperones. Chaperones are the proteins that interact with partly folded or improperly folded polypeptides, facilitating correct folding pathways or providing microenvironments in which folding can occur. So it can do both. One, it can it can directionate your protein that okay, fine, you were unfolded till now, but now you need to go to this pathway. Okay, so it can give a correct pathway to your protein, or it provides a microenvironment such that if your cell is in heat shock, that I've already like multiple times I've told you guys ki if the cell is in stress, then chaperones provide a microenvironment such that this is the outer coating, this is the chaperone molecule, and inside it is present your protein such that it is protected from whatever is happening outside. So this will be your chaperone and this will be your protein. So first line justified. Second says several types of molecular chaperones are found in organisms ranging from bacteria to humans. So what is important is they are present in bacteria and they are present in humans also. They are present in plants as well. Okay, one of my friend is actually working on heat shock proteins only in plants, so they are present in plants also. Next is two major families of chaperones, both well studied, are HSP70 family and the chaperonins. Already I've told you what HSPs as to what chaperones are or what are chaperonins. Okay, both in the form of YT shorts like YouTube shorts or the videos I've uploaded before. In order to understand the practical implications separately, you can refer those videos, and I highly recommend you to go back and watch those videos, because they will give you some ideas to why are there in the cell, why are they there in the cell. Also, I have uploaded one video on Instagram, not to promote all these things, but definitely you'll get a an idea as to chaperones are there when we have fever, because our cell is having heat stress. Okay, so let's move. Now, HSP70 proteins, they bind to regions of unfolded proteins. And where do they bind? This is important for your exam perspective. They bind to hydrophobic residues. They bind to hydrophobic residues of your protein. Okay, so hydrophobic amino acid, say they go and bind the hydrophobic amino acid. Then these chaperones thus protect. What is their additional function? One second. Their additional function is to protect both proteins subject to denaturation by heat and new peptide molecule being synthesized and not yet folded. So I have already discussed your denaturation part. Now let me give you one more thing, that there must be a lot of peptidases. Okay, so peptidases will do what? They will cleave the peptides or they will digest the peptide bond. So in order to protect the protein from both denaturation as well as when the new peptides are being synthesized, to protect them from peptidases, these chaperones are there. Okay, then second function is they block HSP70, which is one kind of your chaperone, also block the folding of certain proteins that must remain folded and that must remain unfolded until they have been translocated across a membrane. So this is a very interesting concept, as to many a times what happens is co-translational translocation. So when there is co-translational translocation, two things take place simultaneously. What are they? One is the synthesis of your polypeptide chain and other is the translocation and the movement of your polypeptide chain into that originally. Say for example, this is your ER. वैसे ER is double membrane, but here just to simplify, I have shown one circle, one big oval structure. Okay, so this is your ER, this is your ribosomes, and this is your polypeptide chain. Okay, it is getting synthesized and at the same time it is getting translocated. So you must have studied about the HSPs or BIPs which are present here, and they bind these protein in unfolded form until it has been translocated into the membrane. What if chaperones were not there? As to what if chaperone will not hold these polypeptide chain, so this protein or the polypeptide chain can misfold, and therefore the organelle in which it is getting transported is has no idea as to what is the proper folding for this protein. Okay, once it went inside this once it goes inside your ER, then only it folds into a particular, or you can say specific structure. So by till that time, the just to protect the protein from getting unfolded, therefore we have chaperones present here. Okay, so one of the function was to protect from heat as well as new peptidases, and second is to block till they get translocated into the other organelles. Some chaperones again also facilitate quaternary assembly of oligomeric proteins. So what are oligomers? More than one subunit. Say for example, this is one subunit and this is second subunit of protein. So this will be an oligomer because it is made up of more than one subunit. And just to facilitate their quaternary assembly, we require what? Chaperone. So these are different functions of chaperones, out of which I think is most important for you guys is this one. They bind to the hydrophobic residues. Then we again talk about, so this is the same slide. So I have just simplified it. It is for binding, and these are the functions of your chaperones. Now in the let's talk about the function of HSP70. So this line again, whatever is important, I have taken only that for you guys. So first of all, let's refer to this picture. See here, it's a protein in unfolded form. This is its folding intermediate, and here it gets formed completely into a native protein. So what can happen when there are folding intermediate, or in at this intermediary stage, is your protein can intermingle and form this kind of aggregate. Now it's a waste. Now it's a waste for our cell, and also it can give rise to certain protein related diseases like prions related disease or amyloid fibers accumulation. Okay, so this is not good for your cell. We do not want this to happen, and just to protect the cell from this aggregation, we have chaperones. Now let's look at the function of these chaperones. So we are talking about HSP70, which is the chaperone. So its structure is one, it has a ATP binding pocket. Okay, when it is bind with ATP, its structure is called an open structure, and it cannot bind anything else when it is already bound with ATP. Okay, so I think this is clear that once it is ATP bound, it's open structure, it can't bind anything else. And it takes help of one more HSP, which is HSP40. HSP40, it does what? It hydrolyzes this ATP and removes the ATP. As a result, the site becomes free and its conformation changes and it becomes a closed structure. So closed HSP70 ADP, and it is a high affinity for what? High affinity for your folding intermediate. Now this folding intermediate can come and bind here, and it will bind it till it gets translocated, or till the system has some stress, till the system is facing some peptidases attack. By that time, their chaperones will hold your protein very tightly inside it. Okay, and what can happen once it happens? It's a reversible process, let me tell you. So what happens once that stress or something has gone out, then again it can convert into its HSP70 ATP, and your protein gets released. How? Here the function of NEF is important, that is nucleotide exchange factor. You all know that just to give protein to something, or phosphate to something, as to whenever we give ATP to something in exchange of ADP, we require these certain nucleotide exchange factors. Okay, so again, ADP here will be given one phosphate by this nucleotide exchange factor. Again, ATP will be attached to it, and HSP70 will now form what? Open structure, and will release the folding intermediates. I hope this is like you understood it. Again, for Hindi medium, I'll tell you what happens is, आपके chaperones क्या करते हैं, वो protein की folding intermediate से bind करते हैं. तो अगर आपकी protein fold हो रही है तो पहले इस form में होगी, देन उसका एक इंटरमीडिएट आएगा और फिर वो नेटिव प्रोटीन बनेगी। अगर इधर पे फोल्डिंग इंटरमीडिएट को प्रोटेक्ट नहीं किया गया तो उनमें एक टेंडेंसी होती है कि वो एग्रीगेट बना सकते हैं। ओके, तो अगर वो एग्रीगेट बना रहे हैं तो सेल की एनर्जी वेस्ट होगी, पहला। दूसरा, सेल में प्रायन से रिलेटेड डिजीजेज हो सकती है मिसफोल्डेड प्रोटीन्स की वजह से, या फिर अमाइलॉइड रिलेटेड जैसे कि अल्जाइमर्स वगैरह होती हैं वो डिजीजेज हो सकती हैं। तो उनसे बचाने के लिए हमारे शेल में शेपेरॉन्स हैं। होता क्या है कि ये फोल्डिंग इंटरमीडिएट या फिर अनफोल्डेड प्रोटीन दोनों बाइंड कर सकती हैं आपके HSP70 के साथ। HSP70 एक तरीके का शेपेरॉन है जो कि इसके पास ATP बाइंडिंग पॉकेट होती है। अगर ये ATP से बाइंड करेगा तो ये आपके किसी भी प्रोटीन से बाइंड नहीं करेगा, तब इसका कॉन्फर्मेशन ओपन कॉन्फर्मेशन बोलते हैं और इसकी एफिनिटी प्रोटीन के लिए बहुत लो होती है। वंस इसको हेल्प मिलती है HSP40 की, वो HSP40 यहाँ से एक PI हाइड्रोलाइज ATP हाइड्रोलाइज करके एक इनऑर्गेनिक फॉस्फेट रिलीज कर देता है। अब आपका HSP70 जो है वो ADP से बाइंड हो जाता है और अब इसकी एफिनिटी ज़्यादा है आपके किसी भी फोल्डिंग इंटरमीडिएट के लिए, या फिर जो आपका मेन प्रोटीन जो फोल्ड होना चाहता था अनफोल्डेड प्रोटीन के लिए। तो अब ये बाइंड कर लेगा और तब तक बांध के रखेगा अपने में जब तक कि सेल से कोई भी स्ट्रेस सिग्नल ना चला जाए, पेप्टाइडेजेस उसकी सराउंडिंग से ना हट जाएं, ताकि आपका प्रोटीन क्लीव ना हो जाए। या फिर अगर ट्रांसलोकेशन हो रहा है, फॉर एग्जाम्पल किसी ऑर्गेनेल में जा रहा है, को ट्रांसलोकेशन हो रहा है, तो जब तक ट्रांसलोकेशन टर्मिनेट नहीं हो जाता तब तक चेपेरॉन उसको पकड़ के रखेंगे ताकि उस प्रोटीन की अनफोल्डिंग ना हो जाए। ओके, ये हो गया। तो अब जब इसको रिलीज होना है, फॉर एग्जांपल टर्मिनेशन हो गया ट्रांसलोकेशन का, पूरी तरीके से प्रोटीन पहुंच गया ऑर्गेनेल में, अब इन शेपेरॉन्स को हटना है। तो उन्हें हटने के लिए कौन हेल्प करेगा? एक न्यूक्लियोटाइड एक्सचेंज फैक्टर होता है। न्यूक्लियोटाइड एक्सचेंज फैक्टर यहाँ ADP है, वापस से इसको फॉस्फेट दे देगा। फॉस्फेट यानी कि ATP और ADP को एक्सचेंज कर लेगा, ADP खुद ले लेगा और ATP दे देगा, जिससे कि HSP70 ATP कॉम्प्लेक्स फिर से बन जाएगा और आपकी प्रोटीन जो है वो रिलीज हो जाएगी इसको जहाँ भी जाना था। अब what is important for you guys is in bacteria HSP70 and HSP40 are called as what? DNA K and DNA J. So DNA K is what? HSP70 and DNA J is HSP40. These are contemporaries. So HSP70 is present in eukaryotes. It's similar in E. coli is known as DNA K, whereas HSP40 is present in eukaryotes and its similar counterpart in your prokaryotes is called as DNA J. So DNA K and J were first identified as proteins required for in vitro replication of certain viral DNA molecules. So these are called as DNA K and DNA J. But इनका interestingly जो डिस्कवरी है वो कब हुआ था? जब DNA मॉलिक्यूल वायरस का जो DNA पार्ट है उसका रेप्लिकेशन हो रहा था, तब इनकी हेल्प की देखी गई थी। एंड देयरफॉर दे आर कॉल्ड एज व्हाट? DNA K as well as DNA J. मूविंग ऑन, एंट्री ऑफ chaperonins इन द पिक्चर. सो लेट्स डिफाइन कि chaperonins कैसे आ गए. Okay, so let's talk about chaperonins. So chaperonins help in the folding of cellular protein that do not fold spontaneously. In E. coli, there are 10 to 15% of cellular proteins which require the resident chaperonin system, which is called as what? GroEL to GroES. So 10 to 15% of cellular proteins are completely dependent on this system, chaperonin system, for their folding. Whenever the temperature is more than 30 degrees centigrade or more than 37 degrees centigrade, sorry, this is talking about above 30%, but whenever the temperature goes up or beyond 37 degrees centigrade, then your GroEL and GroES system is required. Okay, so let's understand this GroEL and GroES system more via a picture. So these are two GroEL chambers and its lid is called as GroES. What happens is your any protein which is a slow folding intermediate or a native protein can come and bind to the apical region of GroEL, and then 7 ATPs are attached to it. Slow ATP attachment is there. Once your protein binds to the apical region of GroEL chamber, GroES binds and the lid closes. Now your protein is trapped inside it, and the 7 ATPs which were attached, now they undergo the hydrolysis. Once they are released, your protein is trapped inside. So what happens is, so whenever this lid will open, your protein can, release and fold into a properly folded manner. Okay, now let's talk about the other half of the GroEL subunit. So what can happen is, you just have seen that there is HSP70 ADP. Okay, so HSP70 ADP plus unfolded protein. This protein folding intermediate can also be delivered by HSP70 ADP to your GroEL system. Look at the down part, it will again will do the same function, but it is helping the chaperone, therefore it is called as chaperonin. Now the folding intermediate can come here, again 7 ATP, again hydrolysis of 7 ATP, and your protein gets folded easily. Okay, So this is what is your chaperonin system. It's called as chaperonin or the assisters of chaperones because of this thing. And the two GroEL compartments occur or act in alternate cycles. Let's read the fact. So fact says that there are family of protein multi-subunit ring. It has two chambers oriented back to back. First one, upper one or exposed chamber is GroEL, and inside this your protein is tapped transiently by GroES lid. GroEL undergoes conformational changes coupled to ATP hydrolysis. ATP hydrolysis is slow and it regulates the binding and release of GroES. Why is your ATP hydrolysis required? Because of the binding and release of GroES, because there is only one lid. This lid can bind to the upper and lower both parts. Therefore your ATP hydrolysis is required to for its movement or for its binding and release. So inside the chamber a protein has about how many seconds? I told you 1 minute, I think. So sorry for that, because I told you 1 minute in the last video. It is 10 seconds only to fold the time required for the bound ATP to hydrolyze. Your ATP will hydrolyze in the meantime. Therefore your protein has just 10 seconds to fold in the proper direction. If your protein is not folded, then it can go back, and if it is unfolded, then it can go back and again it can fold. The ATP hydrolytic cycle is also slower in HSP60 complexes, giving the proteins constrained inside more time to fold. So GroES and GroEL is called in E. coli, and the HSP60 is the same system which we call in eukaryotes. Okay, so last one is my notes. My note says that protein present both in prokaryotes and eukaryotes. So in eukaryotes or plants and animals both come present in cytosol, nucleus as well as organelles. So for example, they are called as HSPs. Their function is to help in the process of folding, assembly of multi-subunit protein, role in translocated or polypeptide. Now you have chaperones. Chaperones mein we have HSP70 family or HSP60 family. They both are chaperones. Mind you, this is just chaperone, not chaperonins in my notes. HSP70, HSP40 or BIP binding protein. They work during early stage. That's why called as early family. Or all are single polypeptides bind with unfolded and they have ATPase activity. So for ATPase activity, they have N to C terminus. N terminus has ATP binding domain. C terminus can bind with your target polypeptide and recognize hydrophobic region of the protein. That's what I told you in the video or the lecture from the Leninger. Okay. So HSP60 family, also called as chaperone. Now HSP60, sorry, yeah, I have a chaperonins in my notes. So HSP60 family, also called as chaperonins, HSP60 or HSP10, they work during later stages because your HSP70 many times ये क्या करता है, ये बाइंड कर लेता है और ये लेके जाता है, बाइंड करता है और लेके जाता है chaperonins के पास, एंड आगे का फंक्शन chaperonins करते हैं. Therefore they work during later stages of polypeptide. मल्टी सबयूनिट प्रोटीन्स होते हैं, ये सिंगल पॉलीपेप्टाइड की बनी थी. मल्टी सबयूनिट अप्रोक्सीमेटली 14 सबयूनिट, 7 ऊपर की 7 नीचे की, इसीलिए 7 ATP हाइड्रोलिसिस होती है और 7 ATP is hydrolyzed because it is made up of 7 upper and 7 lower subunits. Binds to misfolded polypeptide, as I already told you. It helps in the spontaneous folding of those proteins which do not form easily, do not fold easily. So you have GroEL chamber. This is GroES chamber, and they are also called as chaperonins. With this, my lecture ends, guys. Thank you so much, and these videos are helpful for your BRC Biosciences, CSIR, DBT, GATE, whatever one day exam you're going to do. I bet you कि इससे ज़्यादा तो नहीं आएगा, ठीक है? So thank you everybody. Keep studying.